welcome to this last webinar for this year of the SASSB. We're very happy that today we have our past president to give us a lecture, and we're fortunate that this is a lecture that he gave at the Big Botanical Congress, and that he's now willing to share it with us. So I think I do not have to give Muthama any real introduction, but he is at the Bolus Herbarium at the University of Cape Town. I've been working on a lot of genera and places, and will tell us today about what we can do about African flora beyond regional floras. So with that, Muthama, over to you.
Thank you so much, Fanas, and thank you, colleagues, for allowing me a chance to speak to you. I'm being cheeky to give the seminar that I gave at the IBC, but at the IBC, I was slotted to give my keynote on the Saturday at 8:30 in the morning. And there were 3 other sessions which were happening in parallel. So even people who are in Madrid, who are awake, still had to choose from 1 out of 3. And being on the last day after a very busy week, I had a very full room. I think I had 50 people. So I've got more people now, perhaps, per proportion of our population than I had at the IBC. It's a pleasure to talk on a topic which I've attempted to talk about multiple times. Some people have had versions of it. Benny Bitterby and I attended a congress that were invited in Muasya, I think in 2010. And I attempted to do first version of this. Benny, this is version 4 of it. And I'm hoping that at the end of it, at the end of this year, I'll submit a review which captures some of these thoughts. This work builds on... Okay, I need to move to the next, so just allow me to get myself organized. This work builds on a recent special issue of the Journal Annals of Botany, where Jasper Slingsby, Tony Fobuam, and I were the editors leading it, and where a number of our colleagues put in papers which interrogated different aspects of the African flora. This included from work looking at fossil use of phylogenies for lineages in forest, in the open, in the fynbos, looking at ecology, and in total, this body of work, which I believe should still be available to read and perhaps download for free, brings a consensus on our knowledge of the African flora's ecology and evolution, especially looking at the last 60 million years. I must admit that while I'm using that work as a basis for my presentation, any errors that crop up that you see, it's my responsibility, not the responsibilities of the people who contributed to the special issue. My talk today will look at the African flora, and my talk will ask questions on where do we have, what main assemblages do we have, and where do we have them? And I will attempt to use especially fossil and phylogenies to ask a question on their origin and assembly. This is done in the context of our accepting that Africa is fairly poor in fossils. In this work by Jacobs and co-workers, looking at the Miombo woodlands, showed that at the Eocene, there is evidence that the lineages which make the Miombo were there in the fossil. But while they were there, if you look at the families that make them, and the families is the one with the ashes, the families have been around for the lineages that are in the forest, that are in the savanna, that are in the woodland. But the physiognomies, those assemblages that we call forest, are older, going back to Paleocene. Woodlands, Miombo are in Eocene, and the savannas are fossil signature in the Miocene. And then one asks the question, what has happened in Africa? Africa has been fairly stable over time. What has happened that we've had that shift happening? And as a take-home message, I'll say Africa's biodiversity has been shaped by a number of factors, especially a combination of continental drift, a fairly active geomorphological action for past climates and climate changes, and then we've had dispersal and the interaction of the organisms with the biotic and abiotic processes. But let me take you on a quick safari. Everyone knows Africa, but everyone knows, like the people with poor sight, the elephant by what they can touch. Where I am in the Cape, sandstone, 400 million years formation, assembly of the Fembos set, which has assembled in the last 15 million years as the Mediterranean climate has set in. But you go 300 kilometers north and you find yourself within the succulent Karoo. Now we've just gone through the daisy season and the Isoaceae are coming up. Last 5 million, 5 million years, assembly which is arid adapted and rapidly radiating in just a number of lineages. You go to the eastern part of the continent and you've got savannas extensively occurring, and especially if we look at this image from the Amboseli in Kenya looking into the Kilimanjaro, where you have C4 grasslands, where you've got elephants and other animals which have been there for a bit of time. As you climb up from 600 meters to 6 kilometers, you go through an afro-montane forest zone, like the mist forests or the forest around Kirstenbosch, for those in Cape Town, with lineages that have moved and tracked that temperate zone. And then you end up with the temperate alpine with Judd ericas in the snow. And it's in that above tree zone that you get the lobelias. When you're in the Cape, you think of lobelia as a little scruffy annual. Here you get this cabbage-shaped radiation of Judd lobelias and Judd senecio. If we go to the west of the continent, and Africa 60 million years ago would have been covered by pantropical forest, you get multiple layers of forest species, of trees, a fairly rich undergrowth, including distinct species of elephants. But next to the African forest is the Sahel, and then you get into the Sahara. The Sahara's arisen in the last 7 million years. And though Africa's two deserts, the Sahara and the Kalahari is an old cold desert, whereas the Sahara is a recent desert. As we think about Africa and the assembly of African flora, we need to bear in mind drastic shifts in ocean currents. It is thought that up to the early Oligocene, the plates that we see today as the continents, with Eurasia touching into Africa, that were open and sea currents could go from the Indonesian, so Pacific Ocean, through the Wallis Passage above Africa, go through South America, through the Atlantic, between South America and North America, back to the Pacific. And that flow meant that Africa was fairly tropical, and that where we have in the Sahara today would have been perhaps forest. You look at the circulations as we are today, Eurasia and African continents have come together. India has gone up to get onto the Eurasian plate. The Himalayas have arisen. There has been volcanic activity with the closure of the Wallis Channel, and also Northern South America has come together. And that has shifted the ocean currents such that you have in the eastern part of Africa, the Indian Ocean currents circulating with warm water, just coming around the Cape of Agalas and mixing with us cold current, the Benguela current coming from the Southern Ocean, going up to the north. And this enclosure within each ocean system has shifted rainfall patterns, drastically reducing the amount of moisture that's available for plants. And it's thought that especially these became pronounced in the last 8 million years. Well, if you look at on the right hand side, with present going back to 30 million years, we had a period of wetness, high rainfall, up to about 9 million years. And then we went through, or the Africa and the world went through a periods of aridity, and especially the hyper-aridity at about 5 to 3 million years, leading to the collapse of communities in the northern Arabian Peninsula, the Sahara. But this period also coincides with when we get the change happening and in Southern Africa for the West Coast fossil deposits, where we have a shift from a forest community to an open community. So hyper-aridity has affected, perhaps linked with the ocean currents. As a botanist, I'll tell you that it's now the consensus is that Africa has about 65,000 species of plants, not 45. And even though that number is still lower than we have in similar systems in South America, African vegetation is in 6 main vegetation types, with the rainforests around the equator and to the west, even though we've got small pockets, the eastern part of the continent. We've got the temperate Cape. We've got a montane system arising out of the Drakensberg and going all the way to Ethiopia and Socotra, with an alpine at the top. We've got huge savannas surrounding the forest and dissected by the montane. And then we've got the deserts. And that is thought that 60 million years or so, Africa had pantropical forest, and that the emergence of the open habitats has happened in Miocene, with the alpine mountains tops being more recent in the last 10 million years. We are at a fairly privileged time now. We've had floristic treatments, and so my title are two floras. Here I'm using flora in the sense of a treatment of an account of plants, that the oldest one being Flora capensis, going back to about 150 years ago. Flora du Gabon happening, being completed recently, for East Africa. We've also got specimen data becoming increasingly available on African biota, that you can plot my favorite plant, the baobab, and its occurrence within the continent, and you can use those to overlay to see where different species are happening. And it's indeed, it's thought that some parts of Africa are fairly well collected, except when you come into the Sudan, in Central Africa and such. So lots of specimens going back to over 300 years, but not equal effort, to the extent that Africa is thought to have two black spots in the context of Antanellian co-workers, who show that these are areas which are poorly understood but have high taxonomic and diversity and are under threat, the Cape provinces and Madagascar being those dark spots. Now, let's get into the meat of the talk today. The forest, we look at each of those vegetation types, their setting, the ecology and such. The forests, going back to Paleocene, Africa being the ancestral habitat for important groups, including grasses. The ancestor of the Poaceae, who is reconstructed to have been about 60 million years in African forest and diversifying out of the forest, but also importantly, epiphytes and tree species. And if we use the phylogenies of these, we can be able to hypothesize on how the forests have fared over time, and especially work on the Anonaceae, showing that the monodoric clade, work by Dagalia and co-workers in the Thomas Covor lab, showing that Eastern African forest hold the old earlier lineages of the monodori, and that there are three clades: the Eastern Africa with the Central Congolian fragment, and there's a West African fragment of the monodori, and that pattern seems to be matched by a few other forest lineages, to the extent that one could hypothesize that the Eastern African forest fragments host some of the older lineages, and that there has been out of Africa origin of forest lineages. What's unique to Eastern Africa? Let me take the map of Africa and overlay it differently and project especially the formation of the Rift Valley, that this eastern part of the continent, with the uplifts, with the rifting, has had a shift in the landscape, that we have projection with Kilimanjaro going to 6 kilometers, the Ethiopian highlands having extensive areas above 5,000 meters, and surrounded by savannas. And this has caused, together with the southern plateau of the Drakensberg, has caused a shift in the rainfall patterns, but also has offered new niches for species to arise, and has fragmented allopatrically forest lineages in the lowlands in the east part of the continent, the ones in the Congolian basin, and the ones in West Africa. I'm also told, it's hard to believe, that the River Congo flowing to the Atlantic actually was flowing to the Indian Ocean, and that it's the formation of the rifting that shifted the drainage patterns, leading to current flowing patterns. So in the Miocene, we have had formation of Rift Valley, we have had mountains forming, you have outering of precipitation, drainage, and increased acidification. To answer the question of when the Rift Valley formed, there's been a puzzle or a paradox of a whale, a fossil of a whale in the East African Rift Valley around around Lake Turkana, in an area where there's no sea nearby. And this fossil has been dated to about 17 million years. And its explanation is used to understand when the Rift Valley formed, with the thinking that there would have been rivers flowing into the inland, and perhaps this well went from the ocean through the rivers into what is now inland Lake Turkana area and got stranded there. And that with the fossils, it would have gone the River Anza and such, but with the Rift Valley formation, the fossil is stuck somewhere into the west of the Rift Valley where there's no ocean. And that gives an age, a minimum age of when the Rift Valley would have been formed after 17 million years ago. But let's go back to the plants and let's look at a group which is really funky, the staghorn fern, the genus Platyserum. And in the special issue of Annals of Botany that we convened, Zhu and Koakas looked at the origin of this pantropical group, which has got about 6 species in Africa, a lot of species in Southeast Asia. The point that Southeast Asia was thought to be the center of origin. But in making phylogenies of the Platyserum, together with other members of epiphytic ferns, we find that there's a radiation origin of those lineages which are epiphytic, especially the polypoidy, going back to late Paleocene, at a time when perhaps forests were extensive, and that you have the ancestor, the node with Platyserum, the staghorn, is sister to Ovencambia. Ovencambia is East African, and the split between those two lineages is about 35 million years. And then you have a radiation at about 15 million years or so. And these Miocene, mid-Miocene climatic optimum of cooling would have been associated with the fragmentation of the forest. And of course, when you fragment forest and you have epiphytic ferns in the forest, then you give opportunity for speciation to take place. And it's thought that that process led to the increase of number of species within Africa, but also there was a dispersal to Southeast Asia. And in Southeast Asia also there's radiation leading to quite a number of species arising within the last 10, 12 million years. And there's a single dispersal to South America. So using the staghorn fern as an example, we can hypothesize that out of Africa, long distance dispersal, which it's easy to comprehend when you think of ferns, spores being dispersed in the air. But of the Anonesi in the group Bokagi, Mkilua is in East African forest, but the rest of the Bokagi have radiated in the Amazon. Their fruits are fairly large and cannot be blown in the air. Also, they bear their fruits on the stem, so they are not, and their seeds are fairly large, not easily dispersed by birds, so be typically dispersed by primates and such. And to explain the occurrence of Mkilua in East Africa and an African rich fossil, yet South America is where the radiation, Lopez and Koakas hypothesized that perhaps this was a boreotropical land bridge dispersal as opposed to long distance dispersal across the Atlantic. And this explained, especially in that Paleocene period, when Europe was tropical and North America was tropical. And that perhaps would have been a time when the Anonesi would have been in Africa and in Europe and North America. And dispersal over land could have led to that disjunction. That disjunction between African forest lineages and South American lineages is seen in the Maupigiesi, Anonesi, and other lineages. There's still contention. I'm told also some animal groups show the same, so it could be our discussion at the break. But perhaps the most provoking article in that special issue was this by Brown, Lamont, and Koakas, which challenged the currently held or previously held paradigm that the Proteaceae originated in South America, in Australia, and that the group with Protea dispersed into Africa. Yet when they looked at fossil evidence and they interpreted fossil pollen, they show the presence of these pollen which are fairly proteaceous, strongly trigonal, which go back to about 10 million years. And they're in West Africa, into East Africa. And they use this and their absence in Australia to hypothesize that perhaps the Proteoideae group of Proteaceae originated in Africa and dispersed to Australia as opposed to the other direction. The links between Africa and Australia are common and both direction in different lineages. Let's change pace and get out of the forest into the savannas. For my colleagues in Kenya, in Nairobi, you recognize this scene from Nairobi National Park, where you've got a C4-dominated grassland with acacias. Here's the acacia drapanalobium, the whistling fawn, overlooking the city. And very close to this point, Nairobi National Park, you get from savanna with C4 grasses into Afro-montane forest with Olea, Nuxia, and other groups that we know from the Cape Forest. And this being at 1,600 meters is an exciting point to ask the question, where are the savannas, when did they arise, and why? And explanation to this has been attributed to amount of rainfall and the length of dry months, that if you have mesic savannas, where you have more than 800 millimeters of rainfall per year, and you have up to 6 dry months. In such setting, if you exclude herbivores and you exclude fire, you can switch from savanna to forest, or vice versa. On the other hand, we've got arid savannas, which are stable at about where you have rainfall below 500 millimeters or more months of drought. And Africa's these settings that, if you look at the map for Africa, this work by Sankaran and co-workers show that the forests are where it's clear white. In the area which is hashed upright, you could transit the vertical lines, you can transit from forest to savanna, whereas where you have the other grayish, you are more into the stable savannas. And so within the African open habitats, you can get fire-driven systems, the Miombo woodlands being one of those, or C4 grasses, fires regularly coming, or you can have the herbivore-driven system where you have C4 grasses and spiny plants. And fire, spines, and herbivores are good proxies to ask the question on the timing, and we've done this, us and other workers that have contributed to special issue, starting with the work on the spinescence, and this was published, work led by Tristan Charles Dominique, published in PNAS, where we looked at different kinds of spiny plants, and spinescence is very varied. It can vary from the shoots having sharp tips to having stipules, or you can have prickles on that. We treated spines here to exclude palms. Palms have got sharp-tipped leaves, which are fairly different and perhaps evolved under different pressure. If you look at Africa and where the vegetation types, you have in yellow the savannas. If you overlay the occurrence of spiny plants, and in this heat map, the more warm, it's more spiny. You have the spinescence is common in the open. Spinescence also seems to be happening where you have medium-sized mixed social finders. These are the antelopes, as opposed to areas which have got the solitary non-social feeders that we have in the forest. And also these mixed feeders also are coinciding with large browsers and in areas of rainfall, which is not above 800 millimeters. Then you come to the phylogenies. Making a phylogeny of woody plants with a bias on Southern Africa, but sampling into tropical Africa, we find that spinescence has evolved in about 30 or so different lineages, including the legumes. And if we look at the age, when those nodes with spines evolve, we find an age of about 17 million years coming up again and again in the Rubiaceae, in the Fabaceae, in the Apocynaceae and such. What's magical about 17 million years? We took this data and we overlaid it with the phylogeny of bovids including the antelopes. And we find that even though in this figure look at present, going to back about 150 million years, angiosperms have been marching on gradually. But in red, we're showing spiny plants, which have remained poor. At about 40 million years, we got an increase, but an exponential increase at about after 20 million years, coinciding with when the bovids also are radiating. And we explain this to do with the exchange of the biota, with the continental plates coming together, or Eurasia and Africa coming together, and allowing the ancestors of our antelopes to come into Africa to find elephants, the rock hyraxes and such, and to force an arms race towards spinescence. So 25 million years as an exchange. Thereafter, you have radiation of spiny plants. C4 grasses, and I'll not go into the physiology of C4 photosynthesis. C4 photosynthesis is advantageous in warm conditions. It's mostly in grasses, but also in cyperaceae. It's in what we call the savannas. If we look at the timing of the origin, we find that from an ancestor for the grasses in the forest, we have the Pac-Man clade having most of the C4 photosynthesis lineages arising from the Oligocene, youngest in Cyperaceae. So it's not a single origin, it's multiple origin. And that if we look for signal of C4, and I'll use in this work by Eric Edwards and co-workers, I'll use Kenya, and you have a scale from present to the Oligocene, you find that forests are thought to have been in Kenya to about 15 million years, opening up. But as they open up, we're finding that there's signal of a tooth enamel in the teeth of the fossil organisms from about 12, 11 million years or so. So there's a shift to increase of C4 grasses in open habitats in Kenya, in Pakistan, in North America, even in Argentina, which had open habitats all through, but there's increase in C4 after last 10 million years. Moving to fire and fire adapted traits are really interesting. And Frank White, in a paper published in the Singapore Botanic Journal Bulletin of Botanic Garden in 1976, hypothesized what he called the African underground forest. Here's where, for groups which have got trees, like Proteaceae or Gardenia or Cussonia, you have species in those lineages being herbaceous, growing from an underground structure to provide a seasonal shoot, and that flowers and fruits and dies back. And that's as I reasoned multiple times. And Frank White hypothesized that this was driven by fire. And one could ask the question, is underground forest a good proxy? And we did that by using that phylogeny of woody plants and mapping where the underground woody plants are occurring. And we find them occurring in multiple lineages from legumes right to Combretaceae and such. When we ask where they're occurring, we find that there are more towards the equator, and especially around the southern, the coast, but also fairly the younger lineages are as you go towards southern Africa. So older lineages to the equator, younger lineages as you go further south. And indeed, one could use this to say in the opening of the forest, it's at the lower latitudes that you have the oldest lineages and you have younger lineages, or the savannas are younger in southern Africa, in other words. But is it really fire the only leader or driver of this? And in a recent paper by Coutinho and co-workers, using 4 lineages, they tested on whether fire, herbivory, waterlogging, or frost were the drivers. And they find that there are multiple drivers, that underground forests are not occupying a single niche, but they are driven by multiple factors, and that one should not lump it all together, that you have the influence of fire, influence of herbivory, influence of frost, influence of waterlogging, driving different groups differently. But let me get further into the fire story and ask about the Miombo. Miombo and the Miombo woodlands make one of the largest woody savannas in the world. You know, it's a region which fires return multiple times in a decade. And the genus Brachystegia of the legumes related to mopane that we have in southern Africa, and the worm is rather nice to eat, you find that the Miombo have got lineages who are in the forest, and that actually it's hypothesized that from an ancestor in the forest, you have diversification to form the open habitat lineages. And that has happened, that the Miombo species, Brachystegia, have assembled in the last 3 million years. That's a bit frightening. Think Miombo is a system. So my figure that I showed from the fossil, Miombo from Tanzania, they were in the system. They could be picked up from 40 million years, but they were most probably sitting in forest fragments, and that the lineages in the open have assembled rather recently. What about if we go even further, more extreme? Everyone knows these favorite African plants which go around the world and have been given with bad names like mother-in-law's tongue and such. This group of Dracena, the genus Sansevieria, is iconic of the African drylands, and a phylogenetic work published recently shows that the Sansevieria clade has diversified in the last 3.5 million years. So where Sansevierias are, that Somali Maasai system is fairly young also. But amazingly, in the same system, we've got fishes which are annual. I know farmers will say, I'm pulling your leg, that instead of having fish which are long-lived, you have a lineage of fish which the genus Northumbrumcus, which lays the eggs, and the eggs go through the dry season. And when it gets wet, and those wetness could be about a couple of weeks, if not a month or two, they'll hatch, they'll grow rapidly, they'll mate, and the eggs will be the form of dormancy. This group, if you it's more in that Eastern African, East to Northwestern African area. If you put a phylogeny on it, you find that they have an age, Brancus has an age of about 3.5 million years, which coincides with the Sansevieria. Again, using multiple evidence to say, from rainforests, which are pantropical, you have opening, you have Platyserum capturing that fragmentation, you have new niches arising, driven by fire, aridity, or whatever, and then you have lineages which have diversified to go into them. And within this context, perhaps we can attack even the question that non-botanists want to touch about the hominids. You look at the hominids and you ask, where are the oldest ones? Sahelanthropus chatensis from Chad, from about 7 million years. Then you have the Millennium Man from Kenya, or from about 6.6. You have Aridopithecus and Australopithecus coming from 4.5. And the youngest we have is our species. But before then, you have the Southern African lineages. So one could put the plant splits, especially the savanna splits, are shown in black dots, and you could ask where the fossils found. And if you look at a latitudinal gradient, the oldest ones are around the equator, the youngest ones are as you go further from the equator. You're welcome to read this paper, which was published in Scientific Reports. Just see how we were speculating. It took 2 years of being rejected before we could get it published. But then in that open system, we have plants that I can still not put a place to. The pachycaos, plants that have got this unbranched stem, whether it's the baobab or it's members of climbers. You know, this is a relative of the cucumber, which has gone in Socotra, Drontrosicus has grown into this pachycao. And also the yams, the elephant yams also have that pattern. And using the phylogenies from these, one sees that pachycao morphology in the baobabs evolves at least before 10 million years. So it evolves before the C4 systems have evolved. And perhaps this was an alternative route out of the forest into the open lands, which is linked to aridity and which is not linked to fire, even though the elephant yams are adapted to fire. Within the grapes family, Phytaceae, also we see a similar system where the ancestors are vines in forests, and that you have moving from vines to stem succulents, independently happening twice, one group in Madagascar, one group in East Africa, and you have reversal or moving into annuals in different areas. But what about the cycads in that theme on pachycao morphology? Cycads are have been thought to be ancient, except until you get the phylogenies, and in the work by Manka and co-workers shows that they have radiated in the last 10 million years. And when you think of where the cycads are sitting, they are sitting that ecotone between forests and grasslands, where fire comes through, but also frost comes through. And perhaps they are an adaptation of a tree morphology which cannot survive in forests, and maybe that's why they did not diversify when it was only a forest, but have taken up the niche of the opening of forest. Colleagues, allow me to go into the Cape shortly before we finish. I've been talking for about 25, 30 minutes, and the Cape is an amazing place to be. This is the a reconstruction from the West Coast fossil deposit from about 10 million years in an area which today is fairly open, that they had elephants with 4 trunks. The vegetation was forested. And in this death pan, it's been, you can go there. It's amazing to go and see just how much congregation of fossils, whether it was a delta or it was a catastrophe which killed all the animals together. But it's in an area which is fairly barren, that you can barely find a patch, take shade when it's hot. But work done looking at fossil deposits of pollen grains done in the cores in the ocean on a time scale going to about 30 million years shows a dominance of woody kind of forested species, including pollen, which is thought to be from palms, which is Trichomonosuchate reticulate. Pollen of Muasya. We don't think of Muasya in the Cape. We think of them in Namibia. Balanitis dodonea as part of the community before 8 million years. And then you have a shift to the heathy open communities that we have today with Ericaceae, Isoaceae, and Asteraceae. So there's a signature of a shift from perhaps forest to open, and that perhaps coincides with the phases of aridification, going from the starting of the Benguela currents with the closure of these land masses to intensification of it in the last 10 million years, and that 10 to 8 million year period coincides with the image that I showed of the extinctions of aridification in the Arabian Peninsula, and more recently, that process continuing. And looking at plant phylogenies, Rachel Osteen and co-workers looked at 3 groups which are in forest and in the open, starting with the Rutaceae. My colleagues from East Africa will know Calandendron capensis, which occurs in that from montane forest. It occurs in the Cape also, but its sister to the Diosmi. The Diosmi, the Buhus, are in the Fembos and open apidates. They have radiated more recently, but that shift from forest to open is going to about 25 million years. If we looked at the Phylica, and Phylica are in the Rhamnaceae, Rhamnaceae include forest species, the shift from forest to Fembos open is within 15 million years, and in the Pinaceae is even more recent. So we could argue that that period of about 9 million years or so is a period which there was a drastic shift in the plant assemblages. I request my colleagues in the audience who work on animals, do we have similar signature in the springtails or in the cockroaches? Is that when that jumping cockroach got isolated from his relatives in the Cape? But looking at the Cape more widely and asking of the succulent Karoo versus the Fembos, we find that the Fembos in blue has more older lineages, except when you are looking at Pelargonium and Indigofera and a few other lineages, and that the succulent Karoo lineages are more derived out of the Fembos ones. And that also there's niche conservatism involved to explain how we've got the Ericas all dominating, arising out of Europe, using Africa as a stepping stone to arrive and radiating the Cape, or the Proteaceae, whose the Protea sister taxon Pauria is in tropical Africa, but Protea and has radiated into that fire-driven habitat. But working within the succulent Karoo also has shown and some amazing habitats, the quartz fields, which look like landscapes of the moon, fairly looking arid, but repeated in the succulent Karoo in the Namaqualand, repeated in the Little Karoo, repeated in the Overberg, where you have convergence to similar ground-hugging succulent community. And using assemblages from these 3 areas and phylogenies, work led by Daniel Ziegler showed that there has been a convergence and that there's been recruitment from the surrounding matrix, especially in lineages of Isoaceae, Asteraceae, and Crassulaceae, and that from those surrounding communities, you have entry into, for example, the Namaqualand quartz fields in multiple times in the Asteraceae, Isoaceae, Asteraceae, and such. And in a nutshell, if one looks at the Cape and summarizes, as my colleagues Peter Linda and Tony Boam did, we find that the distinct assemblages have had different histories. The deserts, for example, we've got the Kalahari being old, but we've got a second radiation within the succulent Karoo. And that when one looks at those, one needs to think that you could be in a habitat where you're having a signature of different radiations happening side by side. And that conservation threats, especially due to climate change, needs to consider the history and the drivers of the radiation of those lineages. But what's most amazing of being a botanist in the Cape is that you're in a system where you could go from ancient landscapes to recent landscapes, and where fire is an important driver, but also not just fire. Climate also becomes an important driver, like the work of Michael Kramer and co-workers showing that the coldest, especially the coldest month, perhaps has the most influence on the diversity patterns in the Cape, followed by fire and other factors. And there's been exchange between those different communities over time, and there's been exchange between continents, leading to a fairly complex history of the African biota, where Africa has been stable. Africa has stood almost where it is, being tropical, but it has had geomorphological activities leading to new habitats. Aerification has led to new habitats. There's been exchange between Africa and other continents, and such have led to the diversity of life forms, functional trait, functional convergence to occupy particular niches, whether you're talking about the ones in the arid where the Pachycal Dioscorea will occur with the cycads, or we have the Jansenisia and Jan lobelia occurring. And that in this system, we've had the influence of herbivores coming in, the influence of fire coming in, and that together with climate perhaps explains why we've got 65,000 species of plants. I know someone will ask, why is the Amazon richer? Especially when we consider that Amazon was impacted by the asteroid strike, but Amazon has perhaps been a lot more stable, except in the Andes, which also have increased habitats for high elevation arid species. I want to say thank you and leave you with this image from Amboseli again. And I want you to pay attention to what we're looking at, where we're looking at C4 grasses, spiny plants, elephants, antelopes in the savanna. We're moving as you go to 2,000 meters to and from mountain forest. After that, you get into a heathy vegetation. And that's all within a landscape, which the Kilimanjaro is about 3 million years, whereas the elephants have been around for about 30 million years, if not older. Thank you so much, colleagues.
Thank you very much, Muthama, for a very interesting talk. I really enjoyed it. Taking us also a little bit back and explaining some of the diversity that we find here in terms of plants on our continent. So the floor is now open for questions. So if you have a question, just put up your hand and unmute yourself, and then you can have a chat. Any questions? So there's lots of expression of thanks. Anyone wanting to ask a question specifically?
Maybe I'm too intimidating. Maybe it's too clear.
It was so clear that nobody wants to ask a question, but I can't believe that. You can go ahead on the neat. Just unmute yourself and ask the question.
Hi, can you hear me?
Yes, I can hear you clearly.
Yes. Thanks, Muthama. I really enjoyed that. Your map of collection effort showed some dark areas, but also some quite large areas which seem to be quite under-collected. Could you speculate how our perspective on the evolutionary origin and drivers of the African flora might change if we are ever going to be able to fill those gaps?
Thank you, Timo. It's a fascinating question, especially when one looks at where the gaps are, and also when one overlays even the areas that we think we know and the idea of the biodiversity dark spots. So one could ask, why is Central Africa not a dark spot, and why is the Cape or Madagascar being declared a dark spot? And it's definitely, we don't know everything. And for example, in my case, doing field work with my colleagues in the Cape, we find that once you get out of the peninsula, just the Overberg, which is in 150 kilometers, you go into, especially as you go into the Gallas Plain, where you got heterogeneity, you start picking up species which are not even represented in the herbarium by specimens. So yes, we're going to find perhaps Africa perhaps could turn up another quarter. So we could easily go up to 100,000 different species. How would it change our evolutionary understanding? Perhaps we've captured the tips of all branches. That it's unlikely that we'll find something overwhelmingly different, rather than filling in the gaps. That's my perspective for plants, but maybe for animals it could be different. And I've not touched bacteria or fungi where there's even gaps are bigger.
Absolutely. So there's in the question box, there's in the comments, there's a question that say, what are the major unresolved issues?
Thank you. I wish I could unmute everyone and have a discussion on this, because what we're having now, what I've presented to you is a very broad brush explanation. Yet once you start looking at the details of it, you find that, you know, the work that I've presented on the coarse patches, the coarse patches are scattered and they've been understood. They've been, people have gone to them, but you got them if you're lucky and hit when particular species are there. So we have snapshot helicopter based collecting patterns. We've not unraveled the interactions. And for colleagues who are doing pollination biology or any association between organisms, will know that we know very little of that. So I think those are the unresolved gaps. You know, when I was doing my PhD, I was shocked. I was working with a colleague from Europe, working on a group where if I saw a specimen with different morphology, I speculated it could be different. For him, his differences were in measuring the epidermal cell dimensions. Their species are so studied that they know to the population level which one has got a cell ratio of this, and that's different. For us, it's still, you're dealing with one leg of a bee and you use that as the only exemplar you have. So sorry, I've not answered that question effectively.
That's fine. Then Daniel, I think you tried 2 times to ask a question. I don't know if you want to go ahead and still ask your question. If not, you're welcome to just unmute. There's okay. You can go ahead.
So, sorry, Daniel, you might need to type if you cannot.
Yeah, or just unmute. I've asked him to unmute. If you can just unmute. Well, then, if you have battle with that, please just type the question and we will ask it. And then there has been ask about the name of the hominid paper you guys put out, and I think this is the Davies et al. 2020 paper.
Yes, indeed. And
yeah, so we had a response already. In the group, somebody has posted the link to the paper, so thank you very much for that. So Daniel, have you sorted out your microphone issues? I've tried. I cannot unmute him. I just ask him to unmute. Any other questions so long? There's quite a silence. Anybody else that still want to ask Muthama a question, you're welcome. Looks like we've run out of questions. Everybody was happy with the answers and things you've given us, and quite a lot of things to think about, and telling us that there's still a lot of work to be done in Africa, especially filling in those, as you say, the gaps, making it broader. And as you've also indicated, you know, even for the bacteria just associated with these systems, we still have quite a lot to do. Nicola is asking, can you talk a bit more about the pattern of older clades in the north and younger in the south of Africa?
Thank you, Nicola, for that. I talked about the older clades in the context of the savanna, but one could also talk about it in context of the forests. That, you know, that pantropical forest zone, I have no reason to think it did not extend south, so perhaps we've had extinctions. But if one looks at the lineages in the montane forest, for example, they seem to be derived from differently from the ones that are in the forest. And you know, when I think of the forest's lineages, including things like figs, anonesi, and such, those don't get into there from montane system. The nearest you get to them is when you get into the Durban forest system, going up into Hambane. So there seems to be, perhaps one needs to disentangle and say, we cannot say all forest lineages are old. All forests are not the same. The coastal, the lowland forests in Eastern Africa, for example, will contain, and Eastern Africa, I'm saying from Zanzibar to Inhambane and even to Durban, will contain older lineages compared to from the Cape or mixed forest or the more forest in the from mountain system going into Ethiopia. In the savannas, definitely the evidence is that opening up perhaps happened more near the equator, and that's where the older lineages of Africa savanna split for plants are, and also that's where the older hominids are. But is that where the older animals groups are? Maybe we need to use animals to disentangle that. And of course, then you also have the Eastern Arc system, which has perhaps might have a slightly different history. So, Nicola, did I catch your question fully?
Yes, I think so. Thanks, Muthama.
Thank you for that. Daniel has been able to write his question in the chat. So he says, angiosperm diversity is lower in Africa than other parts of the globe, and the aspects of only 2 dark spots in Africa. Do you think Africa has equal sampling opportunities in other botanical exploration as other parts of the world?
Yeah, thank you, Daniel. It's always, especially when we start counting numbers, it's always a question of who is counting, and are they counting the same numbers? Are they counting me, you, and me, adding one more in that? But I've seen more than one study done, especially to ask the question, why Africa compared to South America, for the same land mass, South America has more species than Africa. And I think South America has gaps in sampling, just as Africa, that especially in the Amazon, where you have huge numbers of tree species co-occurring, and also co-occurring at a canopy that you only see the fruits falling or the leaves falling. And if you are slashing the stem to identify species, you cannot go much into that. So perhaps the ecological opportunity in South America and the extinction of African flora, especially the Sahara, might explain the lower numbers compared to South America. The more recent extinction, South America had older extinction lead to the dinosaur, the meteorite strike, but that ushered in the angiosperm diversification, and then South America has had the uplifts in the Andes, and that has created a whole new niche where genera like lupinas and the bromeliads have just gone crazy there. We don't have that. Our high elevation has more the alpine community, but it's not the same level.
Okay, thank you for that. Any last questions you would like to ask? If not, then again, it's my pleasure to say, Nicola, you want to ask another question?
No, I was just switching on the video, so because I know we'll be saying goodbye soon.
Okay.
Thanks.
So thank you very much. It's my pleasure to say thank you to Muthama for sharing this with us. And really, you know, it's really an interesting talk. The people in it was Madrid didn't know what they've missed out on. But maybe the weather was just too nice outside. I just flew in from Madrid 2 days ago, and we still had lovely weather there. But the last announcement I just want to make is we have spoken about starting a phylogenetic series for students, and we will have our first session and start advertising it from next week on the 17th of October at 4 o'clock in the afternoon. But this is really, if you're interested in, and the first topic Martin will deal with is how to read a tree. So we want to really provide some basic information, and we will reach out to the student community to ask their questions. Martin will give a short outline of presentation, and then start dealing with the questions. And it's an open system, so open meeting. So if people are interested in then asking their questions and things, we can deal with it. The people that know how to read the trees are also welcome to join and give their input. So we really want to make this a community effort to start from scratch and build up some knowledge amongst people that use trees but are not that doesn't know it so well. And we really hope that this can be something that we can do, but it's going to be a trial and error. So we're going to see what the response is and whether you like it. It will also be recorded so that we can put it on the YouTube channel. And Abby wants to say something.
Hello, hello. Can you guys hear me at the moment? Sorry, my mic sometimes gives problems. On the subjects of that, I'm just quickly putting in the chat a link to a WhatsApp group. The student reps of SASB have also started like a relatively informal WhatsApp group, but the idea is to have a place that you can ask questions, share student talks, organize thanks, Rob just put the link in, like organize meetups. And next week, Wednesday, in fact, we are having someone present on their workflow of like how they've been processing data from like doing extractions up until getting their results, actually running analyses. And the idea is to basically give everyone a place where you can go and get help outside of your supervisors and reminisce with students who are going through similar things. So if anyone wants to join or pass the link on to their students, please feel free.
Thank you very much. Also to our students reps, who is really doing a lot of work to invigorate the society. We also would like to ask everybody who's on this call, we will send out the link of this phylogenetic talk and question and answer session to the group that we usually send it. Please pass it on to your students and people in your departments and work environments who might be interested in that. This is really for a new generation of people that might be interested in systematics or just would like to use it. So thank you for that. If there's no more comments and things, I just want to say thank you. It was very nice this year again to have these webinars. I've always enjoyed it, and we're looking forward to another few next year, as well as it's time to start announcing dates and things for our next conference, which will probably be in the middle of next year. So with that, keep well and all the best. And thanks for joining. Goodbye. Bye.
Yes, thank you. Before I go, by the way, Muthama, one thing that's really interesting. So you were talking about with the animals. This is only one group, so didn't want to kind of say for everything, but with some of the stoneflies I'm working on, we're finding the same pattern, that the actual lineages in sort of Central Africa, East Africa are quite old. But as you come down to the south, and particularly to South Africa, they all seem to be roughly 3 million years old. This is one of the stonefly groups. So it is definitely a pattern that is happening elsewhere as well, although I can only speak for one group, so I don't know how common it is.
Thank you, Abby. It takes 1 + 1 to see a pattern emerging.
Yes.
Agreed, yes.
Thank you.
Okay, and then one day we will put all the pluses together and write something.
Sure. Bye, everyone.
Keep well. Bye.
Thank you. Bye.